Yeah, I'm just wanting to have a quick chat with you about the origins. Quite a bit has come out in recent months on the origins of COVID. And I'm just wondering, in your mind, what's been the most persuasive evidence?
The best is the most recent paper that came out of Laos, northern Laos. They found a couple of viruses in bats that are among the overall closest to SARS-CoV-2. And most interestingly, the spike protein can bind ACE2 and allow entry of a lentivirus into cells. And in fact, the receptor binding domain, there are 17 contact amino acids between the RBD and ACE2. I think 16 of 17 in one of these isolates is identical to SARS-CoV-2. So that's the closest in terms of spike that we have. And what I think, there's no furin cleavage sites, but this shows that out there are RBDs and SARS-CoV-2 like viruses in bats that are really close to human. They're ready to go. As long as they jump over into people, they can infect them. And then probably they develop a furin cleavage site after a few passages. That would be the idea. So there doesn't have to be an intermediate host. Northern Laos, bats can spread long distances. So I think for many reasons that's really exciting. And it just goes to show we just need to keep doing wildlife surveillance and we're going to find it.
So do you think we're getting kind of closer to getting some important answers?
Oh, I definitely think so. I mean, so what we did with SARS-1, we found all the pieces to make SARS-1 in a cave full of bats, in a certain part of China. We didn't find the proximal ancestor, but we found all the pieces, and we know that these viruses recombine extensively. And I'm sure we're going to find all the pieces for SARS-CoV-2 at some point. This just goes to show not to just look in China. You got to look elsewhere. And, other studies have shown it anywhere from Japan down to Thailand. You can find similar viruses in bats. And the really interesting thing is we never had anything like this in bats before. We didn't do enough sampling. We had SARS-1 like viruses in bats, but never SARS-CoV-2. So now we sample and we're finding them. So I think that's quite exciting. And yes, I do think we're going to eventually find all the bits. It's just a matter of keep looking, right?
Yeah, actually, Bob Gary thinks all the bits are kind of out there now. We've got all the bits in various places. They haven't actually been assembled in a sort of proximal area or something.
Well, I'm sure they're all out there. I'm not sure we found them all yet, but this RBD is clearly one of the bits. And, the rest of the genome still remains to be found, but there are lots of caves and, it's not a trivial task to do sampling. Sampling itself is hard, and to cover all the potential places where it might be is very difficult, but we'll get there, I'm sure.
And it sounds like we've only just begun to kind of scratch the surface in northern Laos, and I don't think we've really looked. I haven't seen much evidence out of Myanmar, but with there's some effort in Cambodia as well. But these have been very undersampled relative to.
Totally undersampled. Most wildlife is undersampled, right? Especially with respect to coronaviruses. So it's not surprising, but we get mired up in these distractions, and then China gets pissed at the world and they don't, they don't want to cooperate. So I think this shows that if you sample, you will find, right?
How important is it that that China is, are going to allow further research within China?
You mean their own research or having other people come in and
well, both.
Things? Yeah, I mean, I think they're not happy about having other people just go in and sample, right? Because, a good fraction of the world has been blaming them. And they said you shouldn't do that, and they get reticent. So um, I think the WHO committee is an example, right? They're supposed to go back for a second round. It's not clear if that's going to happen. It's really up to China. It's up to them to say whether you can come to our country or not. And I think under the current climate, they're not too happy about it. They were more unhappy in the previous U.S. administration, of course, which had a very anti-China stance. This administration is a little bit better, but they still are not perfect, I think. So I think until that loosens up, it's all about diplomacy. You have to fix that.
Yeah,
but within China themselves, they're doing the research, they're sampling. Whether we learn about it, well, we will, because they understand how science works. They have to publish it. So we'll find out what they do for sure. Yeah.
Yeah. How has your thinking about the origins changed over the last year and a half?
Look, I've been a virologist for 40 years, right? Very few people on Earth have thought about viruses as that long. I mean, there are other virologists for sure, but there's not a ton of them. And I have always understood that viruses originate from animals in nature. There have been no lab initiations of major pandemics, with the one exception being, the 1957 influenza pandemic, which looked like it was started during a vaccine trial. Right? So they're deliberately infecting people with that virus for whatever reason. We're not even sure. But this is how viruses emerge. They come from animals. There's just no evidence that supports a lab origin. Now, I understand that you should think about it, but I thought about it and decided that there was far more evidence for a natural origin, which is the case for most other viruses. So I would say from the start, we already went through a SARS-1. We know how that originated. The first time I heard about this, I said, most likely came from some animal somewhere, maybe a live animal market, who knows? But the only thing that I'm shocked about is the fervency of people who, insist that it came from a lab without any evidence. That's kind of surprising to me, frankly.
Yeah, I think it is interesting. If you were to take a straw poll of every virologist who has, knowledge in this area and ask them what they think, I really do think that the consensus would lie in a natural origin spillover event. Yet there is a very vocal minority who are really drumming the lab.
That's true.
Yeah.
Well, I mean, I'm not sure the people who are very vocal are really virologists. You have Richard Ebright and Mark Lipsitch and David Relman.
Jesse
But
Bloom.
they have a history of mistrusting, supposedly risky experiments. They feel that we don't know how to contain them without any evidence for that. And so they jumped right on this one, especially Ebright is very vehement about.
I interviewed Ebright a couple of years ago. Actually, it was when China was working on an African swine fever vaccine, and they were making a lot of progress really fast. And then I looked into how the French had worked with China on building the BSL-4 lab, and how China had these ambitions to have more BSL-4 labs. And I came across something that Richard had said to maybe Science. And I called him up and I had a chat with him, and he was saying that all of these BSL-4 labs in China will be defensive, and that it's all military, and it kind of struck me. I also talked to Bruno Lino, who had a very different take. So it was quite interesting to see his stance, and it really was quite almost extreme.
I mean, it is extreme. That's a good way to characterize it. I just don't see any evidence that, look, the Soviet Union tried for years to make weapons out of viruses. They failed. They're not good weapons. We have other weapons that are far better, and that's what terrorists are going to use. The U.S. had a program. It just doesn't work. So I failed to see, and there's no evidence for it. So, I mean, you can say those things and you get a lot of people stirred up, right? Because it kind of resonates with a certain person, especially here in the U.S. But I don't see any truth to it.
Just going back to the origins, what do you think is sort of missing? What do you want to see, something similar to what was found in Laos, but with a furin cleavage site? Would that be, moving us closer to finding the smoking
Well,
bat?
we might not find that, because that seems to be a human required adaptation, right? So that probably was selected for in the earliest days of transmission. And I don't think we're ever gonna get those samples. Those are probably in the first humans infected, and they're gone. And there's no virus in bats with a furin cleavage site, so we're not going to find that. But the rest of the genome, there's 29,000 bases to account for, which we don't have the proximal ancestor yet. And we have pieces, right? We know some other isolates from bats have a very high RNA polymerase gene identity, for example. So I want to see more of the rest of the genome and how many different recombination events it required. But from the latest paper out of Laos, the sampling done in Laos, they said they can account for most of the genome with 5 of the viruses that have been sampled, including RTG13, a few of the previous RM and Y viruses, and then the 3 that they picked up in Laos. Now, I think there's still more of the genome to be accounted for, and then of course you'd like to know where this happened, because as we were talking about the other day, you can have a bat in a cave in Laos and it flies 10 kilometers, interacts with bats in another cave, gives them their viruses, and then those bats go another, so it could be a whole chain of events. It doesn't have to be that one bat flies 2,000 miles. Bats just don't do that for the most part, right? So I want to see more extensive sampling of other parts of the genome. I don't think we're quite there yet.
And what about the international wildlife trade? Do you think that is a part of the story as well?
It could very well be. We really don't have any idea of what's in bats in other countries, right? They've been, really undersampled for the most part. Hardly anything in the Americas and Europe and Africa. And I'm sure there are coronaviruses in bats, so they could move around by that as well. Sure. I mean, as you can imagine, this is a huge issue that it's a big world, a lot of bats, and especially the species that carry coronaviruses, and all poorly sampled and not a lot of expertise to be able to do that. One of the few organizations that was very good at it is EcoHealth Alliance, and they just get demonized, every day.
Yeah, it feels like that train is not slowing down anytime soon.
They do the right experiments, right? They are trapping, they're looking at genome sequences, they're building models of transmission and movement, and they do it. They don't do it themselves, but in collaboration with North Carolina group and the Wuhan Institute, they ask, are these spikes genes that we find, are they encoding proteins that can bind ACE2? And the answer is yes, as we saw from Laos and we've seen before, for sure.
Vincent, I just wanted you to explain for me the recombination thing. I'm not really familiar with how that works with coronaviruses. I'm familiar with influenza, for example. How does it work with coronavirus?
So it's the same idea. You have to have 2 different viruses infect the same cell, right? So that happens quite often with viruses. And for influenza viruses, as you know, 2 different influenza viruses infect the cell, and they mix their RNA segments, and then the parental viruses have a mixture of RNA segments. It's what we call reassortment. So for recombination, the same idea, you have 2 different coronaviruses. They don't have to be that different. They just have to have changes in different genes. And the RNA polymerase, which is produced in the infected cell, it's one of the viral proteins. It attaches to the very 3' end of the RNA, where the poly A sequence is, starts to copy, makes a complementary strand in preparation for making more virus genomes. And we think that all the RNA synthesis in virus-infected cells occurs in membranous structures that are produced early in infection. So all the RNAs are concentrated on the surface, so they're very close to each other. And if you have two different viruses infecting cells, their RNAs will be near each other. So it's very easy for a polymerase copying one RNA to suddenly switch to another strand, say an RNA from another virus that happens to be nearby it. And the coronaviruses in particular are good at this because the way they make their messenger RNAs is actually by the polymerase copying a bit of the RNA and then detaching and reattaching far away on the same RNA at a certain signal. So that is built into the properties of the polymerase protein. So you can imagine that, detaching and reattaching, it might reattach at another RNA, so to speak. And now you have copied bits of one RNA and another. So that would be a recombinant. So it's pieces of RNA, different RNA sequences actually joined together on the same molecule.
I thought it was interesting, the preprint that came out last week from China, where they had done a lot more sampling in various places of China, and they didn't find anything similar to the SARS-CoV-2 virus, but they did find more coronaviruses in bats.
Yes.
So,
yes.
So that bit of information was helpful.
Well, the more you look, you're going to find more because, as we have said just now, the sampling we've undersampled bats for all kinds of viruses. So you're going to find new coronaviruses. And there was a study not too long ago where they found them in rats, right? They were sampling various rodents, which is another undersampled species. And, rodents are 40% of mammals. I mean, bats are 20%, which sounds impressive, but man, rodents are pretty numerous. They don't fly, but there's a lot of them. And in that study, they found the highest percentage was in rats, and mainly in rats that live near people, like train stations and hotels. So that's really interesting, right? Because they could easily give people the viruses that they carry.
Yeah, a little bit like lepto or something. I'm just thinking of the subway in New York and all the rats you see creeping around.
Oh yeah, that's where I am right now. I'm looking out on 7th Avenue, and whenever I take a subway, there's always a rat walking around, mostly on the tracks, but now and then they're on the actual platform. Yeah.
Yeah. Cool. Last question, Vincent, before I let you go. Thanks so much for your time. What do you think should be the next steps? Where would you like to go with the Origins research?
Oh, I think we need to really expand global sampling, but that is a tough demand because not a lot of groups can do it. Right from the sampling itself, which means, going into caves and knowing how to trap bats and sample them non-destructively, and then bringing the samples in a correct way back to the laboratory, and then doing the right molecular analysis, and then having the right post-sequencing analysis. All of that is very specialized. Not a lot of people know how to do that. I think we need to do more sampling, not just in China and in Asia, but the rest of the world, to really see what's going on. I always tell people, look, we first picked up AIDS in the 80s in Los Angeles, big cities in the US, right? But if we had just looked there, we would never have found the origin. The origin was in Africa, right? Many years before. So you have to open up your mind. And just because SARS-1 began in Guangzhou doesn't mean that this one began in China. It could have started somewhere else. So I'd like to see more extensive sampling in other countries. I've heard a few talks about bat sampling in other countries, and it's always fascinating what they find. So there are bats in Africa that have Nipah-like viruses, like the ones that are in Bangladesh and India, which cause serious human infections from time to time. And we didn't even know that. So I would like to see a greater emphasis on field work. And it's funny because I never did any field work myself. I've talked to a lot of older virologists who have, and who miss, what they call the golden days of field work, where you would go out for a month and in the field, you'd leave your family and just sample for a month and bring back a lot of samples and get some good information out of it. And they said people don't want to do that anymore. People want to stay home and do their work there. So it's a hard thing to get people to do, but somehow, and that's why the lab idea, the lab leak idea is a distraction, because people should be interested in sampling and finding out where it came from. And I think, young people who hear that don't want to do it because they think it's solved, but that's not the case. It's out there and we need to find it.
Yeah. Thank you so much. Are you okay if we use the video?
I'm fine. Yeah. It looks pretty good, probably, right?
Yeah, it does. That's awesome. Thank you so much. Great to meet you.
My
Vincent.
pleasure. Yeah.
All right. Have a great
My
evening.
pleasure. Take care. Bye-bye.
Take care.